In this video, we're going to tackle the problem of how do cells do interesting and ordered, organized things, even though all of their parts are very small and being buffeted around by water. So let's dig into it. So what I'm showing here are a set of videos that are so cool that I have to disappear so you can see them all. What these videos are just a whole bunch of different interesting biological processes. There's links in the description if you want to see exactly what all of them are. But overall, hopefully what you take away from this is cells do amazingly cool ordered stuff. And we talked in the last video that they do this by doing a computation of some sort, right? So what they're doing is they have some internal state that is determined by like what molecules are being made. That determines a set of rules, so which sort of enzyme activities they have, for example, that then updates the state. And so they can do an interesting computation to move around to do all those beautiful things that we just saw by running through this sort of iterative loop. But there's a real problem with this, and the problem is that cells, and even more importantly, their molecules, are very small, and this leads to a problem that cells actually have to overcome. So what I'd like to do in this video is unpack that a little bit. So first, what do we mean by cells are small? So what I'm showing here is a graph on a log scale of length sizes, basically. And it ranges maybe to center us on 0. This is log 10 of 0, which is 1 meter. So this is the 1 meter line. It goes all the way up to millions of meters. And then down here, it goes all the way down to billions of meters, actually 10 billions of meters. And I've annotated this with different objects of the appropriate size. So a cow is about 2 meters, and that's right here on the scale. If we go up to the pando aspen, which is the largest single organism, it's basically a giant tree that went underground to make an entire grove. That's about 8 kilometers across. So the cow is deep in this circle of the pando aspen. Go down to the cow. We've got our cow here. This circle here is circling a very tiny dot, which I'm going to zoom in so you can see. There is, in fact, a tiny dot in there. That tiny dot is the grape. The grape has a tiny dot next to it, which is the grain of sand. So hopefully you're getting a notion for the scale, right? These are all things that as humans, we have a good intuition for because we've all seen cows and we can see grapes. We can walk on sand, that kind of thing. The problem is that life is all happening down here. The biochemistry is all happening down here, where scales get different and harder maybe to intuit. So what I'm adding is I've added a neutrophil, that white blood cell we talked about last time. It's circled right here, so it's about 50 times smaller than the grain of sand. We've got here at the top, this is an E. coli that's circled in there. That's about that much smaller. We go down to next to that E. coli, we've got a viral capsid, so a virus. We go down and look down from there, we can get to an individual protein, and then finally we can get to a proton, an atom on there. These are all way smaller than the cow, for example, and it turns out there's some important implications for that. To get after this, I'd like you to this is one of those points where I'd like you to pause and think about something. Imagine that you are the size of a protein. And if you're the size of a protein, you're this size, how big would a water molecule be? Would it be about the size of dust, about the size of a peppercorn, or about the size of a softball? So pause, unpause. Hopefully you've thought deep thoughts about that. Based on these, it's a rough and ready guess, but it's about the size of softballs, right? If you just imagine this here, it's softball, baseball size around you. So let's actually do that thought experiment. So here you are standing there. You have a water molecule near you that's about a softball size. And actually you're surrounded by water molecules, so now you're surrounded by softballs. And hopefully you've come across in previous things that water doesn't sit still, right? It's bouncing around if it's liquid, so that liquid water is bouncing around. So what we really need to do is imagine yourself surrounded by softballs, dropped into a martini mixer, and vigorously shake it, right? That is the experience of an individual protein that is inside of a cell, just complete random chaos. So how do we reconcile this, right? So if individual molecules are just bouncing around completely randomly, how do we get up to this thing where cells can actually move in an organized, coordinated fashion? To think about that, let's add another dimension. So we were just talking about length scale. Let's talk about time scales now. So this is a time scale graph where seconds are right here in the middle. Again, log base 10. We go up to here. This is millions of seconds. So here's a good party fact. 6 million seconds corresponds to about 12 days of time. And then down here, it gets into much shorter timescales, all the way down to femtoseconds, so 10 to the minus 15 seconds. All the things that I've highlighted here in blue are various protein motions. So here we've got proteins that are moving around. We'll talk in later videos about the exact ways that proteins move, but proteins are flopping around on all these timescales. We've also got different enzyme activities. So these are different sort of how quickly do enzymes catalyze reactions. Those pop up here on the top. And another really handy marker point is actually this. It takes about a millisecond for a molecule of glucose, an important sugar, to diffuse across an E. coli molecule. All pretty fast. Well, how quick are some biological processes? Well, let's choose one, and this is E. coli cell division. So the fastest that you ever see E. coli replicating, like in a lab where it has all the food that it needs, the perfect environment, is about 20 minutes, which means it's up here. And what I'd like to note is that's like 1,000 times slower than these fast protein processes. And in fact, that's kind of an overestimate. So let's actually do another thought experiment here. Let's imagine that we have a metabolic pathway that takes about 100 seconds. So it takes some inputs, glucose, and then energy comes out the backside. Let's imagine it takes about 100 seconds. We have an enzymatic reaction where the actual reaction takes about 1,000th of a second. So obviously much faster than that pathway. And now let's do a thought experiment. Let's imagine that we slow the enzyme down so it takes an entire day instead of a millisecond. So now 1 millisecond corresponds to 1 day. How long does it take to do the metabolic pathway? So I'm going to imagine for a moment you pause and you think about that. So we're pausing. I'm back. Let's think about it. So the way I would do this is I would do this as a ratio. I would say the path rate over the enzyme rate. And so that is going to be 100 over 1 over 1,000, that ratio. And that ratio is then going to be 100,000. So it's 100,000 times slower to do the pathway versus the reaction. What that means is if the enzyme is now 1 day, the path is now 100,000 days, which if you turn it into years is 273 years. So that hopefully gives some intuition for this. An enzyme that's happening once every 1,000th of a second, the metabolic pathway that it's catalyzing is 273 years long. So that's the difference between, that's the difference. So 1 day versus 273 years, that's how much slower the pathway is. What this implies or what this illustrates is this idea of the separation of timescales, which basically says that metabolism and cell biology run thousands to millions of times slower than molecular motions. And that cells can actually overcome the problem that we face here. They average out the randomness by waiting a long time. Let's see, are we, okay, I'm not. Thought for a moment I was covering that screen. Great. Okay, you can average out this by waiting a long period of time. Another way to put this is that cells are playing the long game. Maybe you've heard this in the stock market, that the stock market goes up and down every day. But if you play the long game, if say you bought in 1880 down here, and then you sold in the 2010s, you would have made a ton of money, lots of money, because you averaged over all the random fluctuations in the market, right? By waiting for a long period of time. So what have we seen in this video? We've seen that cells behave in an organized fashion. Cool. But because cells are small, their parts bounce around randomly. And this random bouncing around parts makes it harder for them to do that. They overcome this, however, by doing their organized behavior on a much slower time scale than all these molecular motions, which is known as the separation of timescales. And it turns out as we start going into biochemistry, this is going to happen over and over, where we're trying to understand how some protein works. We're thinking in a very fast time scale, but then to understand how it integrates with the cell, we have to imagine, oh, what happens if this runs forever, basically, and we look at its average behavior. Okay, that is all that I have on this. I look forward to talking to you next, because you might be sitting there saying, well, yeah, but wait, if we're just averaging over randomness, how do we ever get anywhere, right? And so in the next video, we're going to talk about the central bet that cells make that lets them actually go in a direction rather than just sort of fluctuating around randomly. So see you soon.